Thank you for inviting me for presentation. And today, Professor Shyamapada Mandal from Department of Geology, University of Gour Banga, Malda, India. I'm going to present an in silico studies support the anti-scrub typhus activity of the bioactive phytocompounds Andrographolide and Withanolide D. So we know that scrub typhus is a bacterial disease. This is due to the infection of Orientia susugamosi. we know that this is concentrated to in South Asian regions, including India, and currently we have seen their emergence in different parts, including West Bengal and India. And this is an approach for the finding of some alternative compounds which are plant-based, and we have utilized Andrographolide and Withanolide D. This can be available from Kalmeg, that means Andrographis paniculata, the first one, the Andrographolide, and Withanolide D. This is from Ashwagandha plant, so Withania somnifera. And we have targeted a protein of the bacteria Orientia susugamosi, which is known as Orientia susugamosi D ubiquitinase. So before going to the presentation, this is the background. We know that Orientia susugamosi is an obligate intracellular bacterium that cause the scrub typhus, and the antibiotics that are using against its treatment are not working well. So alternative regimens should be there, and this should be effective and without or less with less side effects. And so that we have selected some phytocompounds which are easily available from our locality. And we have seen that for scrub typhus, due to the Orientia susugamosi infection, asymptomatic it may be, or some organs failure occur or death. Reported cases spreading worldwide, and current antibiotics are not always effective, that we have seen. And it is important to understand the molecular mechanism of interaction between lead molecules and druggable proteins from Orientia susugamosi, so that we can target a protein which are important for the pathogenicity or multiplication of Orientia susugamosi in host body, that means in human, and some lead compounds, so that we can produce the lead compounds for future drug. So what is the scope of this study? Molecular docking. This is an in silico approach. Andrographolide, already we have shared that Andrographis paniculata is the plant of Acanthaceae family, and Withanolide D from Withania somnifera family Solanaceae. These two phytocompounds, bioactive phytocompounds, along with the tetracycline that can be utilized against infection of Orientia susugamosi, were utilized as ligands, and the target protein was OTDUB, that means Orientia susugamosi deubiquitinase. We know ubiquitin is responsible for the management of infection caused by the bacteria, such as here Orientia susugamosi. That means this is very important for host body to defend the infection of Orientia susugamosi. And to combat this, Orientia susugamosi and other bacteria are reported to produce an enzyme which is deubiquitinase. This is important to evade the ubiquitin proteasome of human system, so that it can infect and cause pathogenesis in human body. And thus we have targeted this protein OTDUV, and two ligands Andrographolide and Withanolides are utilized. And here ligand preparation is required, protein preparation is required, molecular docking and docked complex 3D and 2D interaction will be analyzed. Lipinski's rule of 5, viability radar, and admit prediction will be determined. So these are the softwares: UCSF Chimera, University of California, San Francisco. This software 1.15, for this study we have utilized 1.16, and that is available from this website, and Pymol for the interaction, as well as the BioVia Discovery Studio for the visualization as well as interaction of protein and the ligands that we have studied in a molecular docking. And open bubble version, this is a software. Without this software, we cannot utilize a file which are utilized in the molecular docking. So in this system, we can convert one file type to another file type, such as SDF to PDB or to PDBQT. And PKCSM as well as Swiss Admi, these are two web server. With this web server, we can determine the admit properties as well as the oral bioavailability and the pharmacokinetic properties of the ligands to be utilized as drugs in future. And RCSB PDB, this is the bank, protein data bank. And from this protein data bank, we have retrieved the 3D structure of the protein OT, ubiquitinized. And PubChem, this is a bank of small molecules. That means the ligands such as Andrographolide, Withanolide D, and tetracycline, we have retrieved 3D structure of the components. And these are the structures after optimization. We have optimized the structure after downloading from the PubChem by the utilization of Chimera, which can be available in the internet. This is a web tool. So this is the structure, 3D structure of Andrographolide. This is very important for the inhibition of viral growth or the infection of bacteria, fungus, as well as it is an immune boosting compound. And like Andrographolide, Withanolide D is also immune boosting, and it has role against viral inhibition as well as anti-cancer activity. And tetracycline, it is an antibacterial agent that can inhibit the 30S ribosome for their function, so that it is a very good agent to kill gram-negative bacteria actually. And here we have utilized as a experimental agent as a standard tetracycline, along with the experimentation of Andrographolide and Withanolide in Silico system. This is the protein structure. This has been downloaded from the PDB protein data bank, and its ID is 6UPU. So with this ID, we can retrieve from protein data bank 3D structure of the protein data bank. And this is the Orientia Susugomosi OT DUV. And this is the structure of X-ray diffraction, and resolution is 2.2 Armstrong. So this is a very good structure. And after optimization in RCS in Chimera, it is prepared as a target protein by the removal of the ligand if it is attached before, and the ions or the other solvents are removed. So this is the prepared protein and ready for docking. And before molecular docking, we have authenticated the structure of the protein through 3D 1D verify. And here we are seeing that 91.27% of the amino acid residue of the protein OTDUV have a score 3D 1D score which is less than greater than or equal to 0.2, and it is passed when the amino acid residue at least 80% scored such values. That means greater than or equal to 0.2. So this is a very good protein structures in 3D 1D justification. It can be done in ProCheck. So using the ProCheck tool, we can prepare the Ramachandran plot of the protein target OTDUV, Orientia Susugomosi deubiquitized protein. We are seeing that 92.6% amino acid residues are located within the favored regions. And we know that when the number of amino acid residues are located at least 90%, then it is a very good structure for the utilization in molecular docking. And we are seeing that only 6.9% amino acids are located in additional allowed region, and there is no amino acid residues in this allowed region. That means in the white region, and the red region is the most favored region. The deep yellow is the additional allowed region, and generously allowed regions are the light yellow. So in the light yellow region, only 9 amino acids are present, and overall 92% amino acids are located in the most favored region. So this is a very good 3D 1D structure of the protein along for the molecular docking study. So before molecular docking study, we have determined the grid box using the UCSF Chimera tool. So this is the website. 1.16 version we have utilized, and dimension we have prepared. This is the dimension of the protein for the molecular docking of a ligand to be docked with the protein, and center was like this. This is in Armstrong. So after docking, we have seen that the Orencia succinylcholine deubiquitinase enzyme with Andrographolide. This is 3D structure and this is 2D structure. Here we are seeing that these are the amino acids. 6 amino acids are interacted with the ligand. This is the 3D structure of ligand Andrographolide. And here this is 2D structure. This is Andrographolide, and the amino acid residues are presented here. The green color presents the conventional hydrogen bond. Carbon hydrogen bonds are asparagine, and other bonds are alkali. So different bonds are available in the interaction between ligand Andrographolide and OTDUV, and the affinity was very fine because we got the binding energy minus 8.4 kilocalorie per mole. We know that when the binding energy in the interaction between a protein and ligand is less than or equal to minus 6.5, then the affinity of the ligand towards the protein is very good, and the binding is tight and interaction is better. So this is a very good ligand, Andrographolide, and interacted very tightly with the protein. So the inhibition was strong, created by the Andrographolide against the protein deubiquitinase from Orencia succinylcholine. And these are the 8 different types of interacting bonds that are formed between the Andrographolide and the target protein. And this is the another molecule, small molecule, that means the phytocompound Withanolide that can be extracted from the Somanifera Withania. And here 7 interacting amino acid residues are available that are taking part in the formation of different kinds of bond with the ligand. This is the 3D structure of Withanolide, and this is 2D structure. These are the amino acids in spike form, and the binding energy here is very nice compared to the Andrographolide, which is minus 10.2. So this is a very good component to inhibit the Orencia succinylcholine deubiquitinase enzyme. So inhibition property is better compared to the Withanolide. So Withanolide can be selected for drug preparation. And these are the various bonds that are developed during interaction between the ligand and protein, OTDUB and Withanolide D. 11 such kinds of number of interacting bonds are formed. And this is the standard drug, tetracycline. This is an antibiotic that can inhibit the 30th ribosome of bacteria so that it cannot synthesize protein, and the killing of bacteria occurs. And here we are seeing that 6 amino acid residues of the protein DUV have taken part to form different types of bonds with the ligand. This is tetracycline 3D structure, this is 2D structure, and here attractive charge with asparagine 39, and in green color arginine, tyrosine, and aspartic acid. And these are responsible for the formation of conventional hydrogen bond and carbon hydrogen bond. And alkaline and pi-alkaline bonds are available with lysine and protein against the tetracycline. So due to the formation of different kinds of bonds, the binding energy available in this interaction was minus 9.1 kilocalorie per mole. And these are 10 number of interacting bonds that are available in the interaction between DUV and tetracycline. And pharmacokinetic properties analysis of the ligand, Swiss ADME and PKCM tools are utilized that we have always Lipinski's rule where performed bioavailability radar was prepared and the ADME analysis were done. So this is the Andrographolide for which we have determined the PKCM tool. Here we are seeing that lipophilicity, size, polarity, solubility, flexibility, and saturation, all the values are located within the pink region of the bioavailability radar. So this is a very good component. And here Lipinski rule, there is no violation because the molecular weight of this compound is less than 500, and number of donor hydrogen bond is 10, and acceptor is 10, and less than 10, and donor is less than 5, and its molar refractivity is 95.21. So this is a very good compound obeying the Lipinski rule of 5 without any violation. And this is admit profile. We are seeing that there is no skin permeability, and AIMS toxicity is negative, as well as there is no hepatotoxicity and skin sensitization. And the oral rat acute toxicity value is so high. Also, the oral rat chronic toxicity is high. And the Withanolide D, this is a very good component that showed very low binding energy, which was minus 10.2 kilocalorie per mole. In this case, also the lipophilicity, size, polarity, solubility, flexibility values are located within the pink region, and its bioavailability ratio is like the Andrographolide, which is bioavailability score is 0.55, and there was no violation in Lipinski's rule. So this is a compound that obeyed the Lipinski rule of 5. So this is a very good oral drug. And in case of admit profile, we are saying that the AIMS toxicity is negative, hepatotoxicity is negative, skin sensitization is negative. And the oral rat acute toxicity LD50 and oral rat chronic toxicity LOAEL value are high. So this is a very good compound. And the standard drug tetracycline, we are seeing that only polarity, that means the topological polar surface area, this value is high, which is greater than 130 Armstrong per Armstrong square. Still, it is obeying the Lipinski rule only with one violation, which is for the donor hydrogen number, which is greater than 5. And there was thus one violation, and the bioavailability score is 0.11. So this is not a good oral drug. It can be used as a injection, and its admit profile, this is a known drug. This is already are utilized against various bacterial infection, and you are saying that AIMS toxicity is negative, hepatotoxicity is negative, and skin sensitization is negative. So that in comparison, we are seeing that like tetracycline, Andrographolide, and Withanolide D showing the same admit profile. So the Andrographolide and Withanolide D can be utilized for the inhibition of Orientia susugomosi. And this is TPSA, topological polar surface area, and W log P value. These are plotted in x-axis and W log, and this is TPSA. So we are seeing that in boiled egg model analysis, in the white region, Withanolide D and Andrographolide are located. So this indicates that these two compounds available from the two plants, different types of different family, are very actively absorbed after oral consumption in our intestine. Whereas this compound, this is actually the tetracycline. This is located outside the yellow region as well as the white region. So its intestinal absorption is poor, and no compound was located within the yellow region. So these all are blood-brain barrier negative, and PGP positive Withanolide D as well as Andrographolide D along with the tetracycline because of blue in color. And lastly, the conclusions. Molecular docking authenticated the inhibition of OTDUB protein by Andrographolide and Withanolide. These are two bioactive phytocompounds that are respectively extracted from the Andrographis paniculata, a Ayurvedic medicinal plant, and another Ayurvedic medicinal plant, Withania somnifera. Both the phytochemicals, Andrographolide and Withanolide D, had good affinity to the target protein OTDUB, and these are very good inhibitory agent against the target protein Orientia susugomosi D ubiquitalized. Stable protein ligand complex formation was due to high binding affinities of the ligands. Andrographolide binding energy is minus 8.4 kilocalorie per mole, Withanolide 10.2 kilocalorie per mole. Whereas we are seeing that tetracycline had the better activity compared to the Andrographis, but Withanolide D was better to inhibit the OTDUB compared to the tetracycline. So here, Withanolide D was found very effective against the target protein OTDUB. Admit profile signify the usefulness of the phytochemicals against Orientia susugomosi infection causing scrub typhus. So here both Withanolide D and Withanolide Andrographolide, both are very good lead compounds to be utilized for the preparation of alternative agents for the inhibition of OT infection in human. But the Withanolide D, due to its very low binding energy in the interaction between Withanolide and the OTDUB protein, so it is a selective one and can be prepared as agent for the treatment. But before that, in vitro and in vivo experimentation is required, and clinical experimentation is also required. So lastly, I acknowledge second edition of Euro Global Conference on Biotechnology and Bioengineering virtual event 2022 organizing committee organized by Magnus Group for the recognition of my research and supporting me for presentation. Thank you. Stay safe, stay healthy. Thank you.